Okay, thanks, Ina. And thanks, Albert, for organising this meeting. I think it's a really great opportunity for me as a basic scientist who has recently started to interact with members of the biopharma industrial community to see how important the next steps in terms of developing some of the basic observations we made are for commercial application. So really what I want to talk about, and I'll keep this brief, I know that the size of or the number of speakers in the session has increased, is a very simple story actually. It's a straightforward story. It's based on an observation and a discovery that we made in the lab, which wasn't directed towards any sort of commercialization at the end of the day, which I think Ina alluded to. It is very important to maintain a funding structure for basic science, for blue skies research, as it were. But it is a program which we've developed into a potential new therapeutic angle in the context of inflammatory bowel disease. So I'm starting off the talk with this very sort of dramatic, this is not the Wicklow Mountains, this is Mount Everest. And the reason I'm putting this across is because I want to get across the concept of adaptation. We have an innate powerful capacity to adapt to low oxygen concentrations and to induce in a protective response, which allows us to then deal with further exposures to this environment. So I'll talk about hypoxia during the talk. Hypoxia refers to low oxygen concentrations. So you all know that if we were to be taken immediately from here, sea level in Dublin, to the top of Mount Everest, where the oxygen levels are about 10%, most of us would suffer severe pathology and ultimately most of us would actually die from it. This is not what mountain climbers do. They go to base camp, they allow their body to acclimatize, to switch on these adaptive responses which help them then to deal with further exposure to low oxygen concentrations. It's a beautiful and elegant adaptive response to this stress. And really the nub of what I want to talk to you about today is a hypothesis that we came up with a few years ago, where we hypothesized that if we could pharmacologically activate this response, trick a tissue or an animal into thinking it's oxygen deprived or hypoxic, could we switch on this adaptive response ultimately for therapeutic benefit? And that's really hopefully what I'll be able to convince you of during the next few slides. So first of all, we're talking about oxygen. I need to get across the point to you that oxygen is really, really important. I don't advocate putting a plastic bag over your head to get this point across, but I think most of us know what happens if you do. And the reason for that is that oxygen has been a really key driver of human evolution since the very, very beginning. So even though the Earth is about 4.5 billion years old, it's relatively recently that oxygen levels have started to accumulate within the atmosphere of the planet. And it's actually that presence of oxygen and our ability to use oxygen for metabolic processes, such as generating the energy we needed to be to go from being bacteria to multicellular organisms, was provided by this oxygen. So oxygen plays an absolutely essential role in providing the energy we need to survive. Now, with that in mind, it maybe isn't that surprising that over the course of our human evolution, we've developed the ability to respond to a drop in oxygen concentration, respond to this hypoxic stress at the molecular level to allow us to increase the response. Now, I'm going to give you a little bit of background science here. It's not too complex, but it's actually really fascinating and an area of exploding importance from a therapeutic point of view. And it's centered around a protein that accumulates in cells called HIF-1, or the hypoxia-inducible factor. Now, in normal circumstances, HIF-1 in the cell is immediately degraded. It can't do anything at all because as soon as it's made, it gets broken down. And that breakdown is dependent upon the availability of oxygen. So you're all sitting there, you're perfectly well oxygenated, and the HIF that gets made in the cells, every cell of your body makes this, by the way, but it's immediately degraded because you don't need it. There's oxygen around. Now, if you go to the top of Mount Everest, or if you block the oxygen supply to a tissue by decreasing the blood flow to that tissue, which is much more important from a medical point of view. These are many diseases, cardiovascular disease, cancer, chronic inflammation, where blood flow is decreased to a tissue. These enzymes, these PHDs that were previously responsible for breaking down the HIF, no longer work because they don't have the oxygen available to fuel them. And in this situation, this protein HIF accumulates in the cell, and it switches on a whole load of proteins. The cell starts to express a whole load of proteins that are directed towards getting oxygen to that tissue as quickly as possible. So what are these kind of things? Well, there are things that grow new blood vessels in the tissue. There are many examples of this. I won't go into them, but they're called angiogenic factors. They'll help the tissue to grow more blood vessels. To vasodilate the vessels that are there, make them wider, increase the blood and oxygen supply to that tissue. And also, actually probably the classic one was in the newspapers a lot recently, Lance Armstrong's favourite recreational drug, erythropoietin, EPO. We actually have the capacity to generate our own EPO, increase our red blood cell mass, and thus increase oxygen oxygenation of the blood system. So this beautiful, simple pathway which activates this protein leads to the activation of these genes which increase the oxygen supply and consequently help that cell to adapt, to deal with the hypoxic stress that it's been exposed to. So under these conditions, any conditions where you get exposed to a state of hypoxia, activation of this pathway is ultimately protective or adaptive. So again, just to reiterate, the main point that I want to get across to you today is that if we can mimic this response pharmacologically or pharmaceutically, can we induce a protective or adaptive response in disease processes? So just to summarize it, I'm going to keep it straightforward so I don't lose people in terms of the science, but hypoxia, when oxygen levels are deprived, leads to an inhibition of these enzymes called PHDs, and that leads to an activation of HIF, and that leads to adaptive gene expression, which ultimately we hypothesize is of benefit to the patient. It's certainly of benefit if you go to the top of Mount Everest because it helps you to increase oxygen supply in terms of the whole body. The other key point, and this is something in the field which has developed very recently and very importantly, is that we don't need oxygen deprivation or hypoxia to activate this pathway. It can be activated pharmacologically, and we and others have been involved in the development of a range of new drugs which are at the experimental stage now, called prolyl hydroxylase inhibitors, or PHIs, which allow us to pharmacologically block these enzymes and essentially trick the cell, the tissue, and the animal, or the human, into thinking it's hypoxic when it's not, to activate this adaptive pathway. So we've become interested in the potentially utilizing this approach in the context of inflammation. Now, there's a lot of background information we could go into here, which we don't have time to at the moment, but as many of you are aware, chronic inflammatory disease is a major clinical burden in the world. These include diseases such as inflammatory bowel disease, which I'm going to focus on for the remainder of this talk, but also, as David alluded to earlier on, arthritis, psoriasis, atherosclerosis, many of the key prevalent debilitating diseases which are currently experienced by man are actually based in an inflammatory process. That is, an inflammatory process that is not being appropriately controlled in the body. The other point is that for many of these diseases, such as IBD, psoriasis, and atherosclerosis, and indeed arthritis, the current therapeutic regimen is suboptimal. There are still patients, as many of us will know, suffering from things like Crohn's disease, ulcerative colitis, and arthritis without developing maximum potential therapeutic benefits. So these are diseases, as David alluded to earlier on again, which are in need of therapeutic development. So the one that I'm going to focus on, we've heard a little bit about already, is inflammatory bowel disease. This is what a healthy colon looks like from an endoscopic image. You can see it's got a nice shiny wall and a clear sort of mucosal surface. A patient suffering from inflammatory bowel disease, in this case ulcerative colitis, suffers as a result of a breakdown of the barrier of the gut from a chronic inflammatory process, which is extremely malignant and prevalent. The breakdown in the epithelial barrier leads to inflammation, which causes further breakdown in the barrier, and ultimately this results in this chronic cyclical inflammation, which underpins these diseases like Crohn's disease and ulcerative colitis. It's a prevalent disease. At any one time, more than 1 million Americans, and they're the people with the best statistics on this, are suffering from some form of inflammatory bowel disease. And again, it is really therapeutically poorly serviced. So people are still getting very high doses of steroids. More recently, the development of biologics, antibodies such as those against TNF have been developed and have greatly improved therapeutics in the disease. But there are many problems. There are many non-responders to these drugs, probably somewhere in the region of 50%, and many people who are initially responsive lose their responsiveness. So again, inflammatory bowel disease is a disease which is prevalent, it's severe, and it's badly in need of new therapeutic approaches. So this is the disease that we chose to further examine in terms of our hypothesis. So again, we spent about probably 7 or 8 years trying to carry out experiments where we were investigating the impact of these hypoxia mimetic drugs upon inflammatory bowel disease. And all of that work can really be described in this slide, which hasn't projected as nicely as I would hope it would, but I think it gets the point across. And that is that if we use a mouse model of inflammatory bowel disease, we can induce a profoundly protective response in those animals. This is what a healthy colon looks like for an animal. So in this case, we're cutting a cross section through the colon. The middle white bit is where the food would be passing through. And here you can see what to a gastroenterologist looks like a healthy mucosal surface. You've got these nice undulating mucosal processes which look like a healthy gut. If we give inflammatory bowel disease to the animal by breaking down the epithelial barrier, there's a complete loss of architecture. There's a lot of accumulation of fluid. This is called edema. It's just a cardinal sign of inflammation. And all these little black spots are inflammatory cells which have made their way into that tissue and are underpinning this inflammation that drives the disease. If we treat mice with a prolyl hydroxylase inhibitor, so remember here what we're doing is we're tricking the mouse into thinking it's hypoxic by introducing this pharmacological inhibitor of these PHD enzymes, we can see that it may not be perfect, but it's a hell of a lot closer to the healthy than it is to the disease. And this is reflected when we look at a number of clinical readouts for those mice, such as their weight. This is a very simple, straightforward, robust, but accurate reflection of disease progression. So if we take an ordinary mouse that hasn't been treated with DMOG, which is our pharmacological inhibitor, I should say, that's the name of the drug, the PHI, which we're using to block these inhibitors and to block these enzymes and mimic hypoxia. What you see is an untreated mouse, when we give it the stimulus for colitis, it loses weight. The animals are getting sick. They're developing all the symptoms that are reflected by the histology I just showed you. And over a course of time, they lose up to almost 10% of their body weight. So these animals get quite sick. Those animals which were pre-treated with the hydroxylase inhibitor are profoundly protective, and that's reflected by a lack of weight loss. We can also measure disease activity index, and this is a composite score of diarrhea, blood in the stool, and other clinical symptoms of inflammatory bowel disease. And again, what we see is the untreated mice shown up here develop disease. The activity index is a reflection of how severe the symptoms are, and those animals shown in red here that are pre-treated with this hypoxia mimetic, this DMOG drug, this prolyl hydroxylase inhibitor, are again profoundly protected. They're almost indistinguishable from the untreated animals when we look at this. So this again gave us great hope that this hypothesis of mimicking a hypoxic response, tricking the tissue into thinking it's hypoxic and it's not, is effective in producing a therapeutically effective effect. Now, one of the most important things in science, as many of us will know, is reproducibility. And importantly, a number of other groups have subsequently published work using different models of inflammation in the intestine to sort of securely demonstrate that this wasn't just something that we found in the lab in Dublin, but was something that was demonstrated multiple times in many labs around the world, which again, I think really profoundly supports the idea of using these hydroxylase inhibitors in the treatment of inflammatory bowel disease. So at this stage, we're very optimistic and enthusiastic about the potential usefulness of these drugs in IBD. One of the problems of the drugs, however, and I alluded to this earlier on, and this is really where our interaction with biopharma industry came, was when we went to conferences and we present this work and we would demonstrate this protective effect of the drug in the context of the intestine, and people say, well, that's great, but the problem is the whole animal is going to think it's hypoxic. So it's going to produce all sorts of other adaptive responses, like an increase in erythropoietin. Now, if you're Lance Armstrong or somebody else who is perfectly healthy and wants to get their red blood cells up, that's actually a very bad thing. It's going to increase your risk of cardiovascular effects, disease. In anemia, it may be a useful thing, but in inflammatory bowel disease, it's not something you would want. So there are a number of unwanted side effects that would occur if we just gave a systemic dose of this drug. Basically, we would like to target it to the intestine and to get it released in low concentrations locally, which would be effective. Therefore, it would be beneficial to develop a formulation of these hydroxylases, which could be targeted, the hydroxylase inhibitors, which could be targeted to regions of inflammation. And this is where our interaction with Sigmoid, through interactions with the CEO, Ivan Coulter, who I think at the moment is en route from Japan, took place. I won't go into this because Monica's already spoken about it very nicely, but I think quite clearly what we were interested in doing is formulating DMOG into these delivery capsules in a way which would enhance their deliverability, if you like, to the intestine. So I'm just going to show one piece of data before I finish up on this, just to demonstrate the utilization, or the utilizability of this approach. Again, we're looking at disease activity index in mice. This is what colitis in a mouse that's not treated with any protective hydroxylase inhibitor. This is the protective effect I showed you earlier on when we just give it systemically. So these mice are protected, but they've also got all the bad stuff, the side effects that we wouldn't want to see. If we just give the DMOG orally, as in an oral delivery, so here we're giving it intraperitoneally to get a systemic delivery, it's completely ineffective. So obviously it gets broken down in the stomach. It's not giving any protective effect. But if we give an oral dose of DMOG, this prolyl hydroxylase inhibitor, in the formulated approach, we get almost the same degree of protection as we do with systemic administration. And quite importantly here, we're using about an 8-fold lower total dose than we have to give with the systemic administration. So the bottom line is that oral targeted delivery using the LEDS formulation is greatly enhances the protectiveness of this drug and at the same time reduces the side effects. So I just want to finish there here, the members of the lab who've taken part in this. Pádraig Fallon in Trinity has been a great help in terms of the in vivo models. I've already mentioned Sigmoid, Ivan, Monica and all the team there who've been an absolutely fantastic help. We've been consistently and for the last 10 years been funded by SFI, and we've also received funding from ERCSET for a PhD and now a postdoc. And I'd really reiterate what I said at the beginning, that I think this interaction between industry, academia, is absolutely of key importance. I don't think I have to convince you of that, but I do think we have to be careful in terms of the funding opportunities that come up, that they're not completely restrictive to applied science at the expense of blue skies research, which I think quite often throws up these sort of interesting stories. So I'll leave it there and talk to you at the end. Thanks.